Thank you very much, Chris.
Okay, well, thanks very much, Julie, for the introduction and for inviting me to present a talk in Fungisphere. Can you see my slide, my screen, share screen? Good.
Yeah,
okay,
yes.
So let me start my talk now. So this is the topic, fungal spores. This is
Chris,
a work.
This is presenter view.
Sorry?
This is presenter's view.
Okay,
We are
so
seeing presenter's view. Yeah.
you're seeing that, so let me get it fixed. Hold on. Sharing. And let me see. What about this one? How about it now? Is it good or still no good?
It's jumped back to presenter view.
Okay.
But look, it's okay with me. I don't know if that's okay with everyone else. I can still see the screen quite well.
Okay, let me try this again, and I will swap the screen. Now, how's that? Is that working?
That's better.
Okay, so let me pull out my pointer. Okay, yeah. So this is the topic that I'm going to share with you, and I'm going to present experimental results to convince you that fungal spores are actually not so dormant at the stage of development, and we also present results to show you when they actually start to go into dormancy. And in a way, this really makes us wonder what fungal dormancy really is. And so before that, I would like to briefly introduce University of Macau. It is established about 40 years ago, and we actually moved to a new campus that is 20 times larger than the old campus in about 2014, a year after I joined University of Macau. And I joined mainly because they established a Faculty of Health Science department, and I was among one of the first people who joined the faculty. And when I first joined, the buildings were brand new, and the labs are all just empty space, and we have to fill up every single thing with new equipments, new benches, etc., etc. And it took us, and you can see this is taken at that time, and you can see the renovations are still going on. And so since then, we have actually established quite well, and this is our building, Faculty of Health Sciences, and this is my office space, and now my lab space as well. I used to be in another place, a temporary space for a few years. Anyway, for those who are not aware, Macau is actually famous not for research, not for university, but for casinos. And there are 35 casinos in a very tiny space of Macau. Macau is only about 30 kilometers square, and we have 35 casinos, and this is only a few of them shown here. And so when you walk around Macau, and you will see casinos everywhere. And so a bit about my research, and Julie has already started introducing my lab. And my main interest since I was an undergraduate is about transcription and gene expression or gene regulation. And so I haven't changed since, and I'm really interested in how genes are controlled. And I studied this in Aspergillus nidulan when I was PhD, and then and Saccharomyces. And in Kevin's lab, mostly focusing on the core machineries, transcription, how they initiate, and after initiation, what happens to the machineries to the end of mRNA being made. And so when I started my PhD, my independent lab, I really wanted to apply what I learned to understand fungal physiologies. And so my lab currently is somewhat not very focused in a way, because I look at many different fungal physiologies, but we are also focused because every questions that we ask are coming up from transcription regulation, and everything really needs transcription to sort of give information. And so I don't think I need to spend too much time to tell that a lot of transcription regulatory mechanisms are first understood in simple eukaryotic organisms like Saccharomyces cerevisiae and Schizosaccharomyces pombe. And a lot of insights has been sort of translated into understanding human or higher eukaryotes transcription regulation. And so when I sort of worked in Kevin Stru's lab, and I thought, well, multicellular fungi, like filamentous fungi, like Aspergillus nidulans, maybe actually better models. They can actually provide additional insights in addition to Saccharomyces because they are multicellular and they have different, very well-defined cell types and very well-defined developmental processes. And so when I got a chance to establish my lab, I thought, okay, why not I combine what I learned in my postdoc and then apply it to what I use in my PhD, which is filamentous fungi, Aspergillus. And also because Aspergillus has some infection related aspects, and that makes my research more relevant. So I want to give a little bit of introduction to fungal genome organization and how transcription is controlled. The organization and transcription control is relatively simpler in fungi compared to higher eukaryotes. Most of the time, the promoters is usually within about 500 base pair of a gene. And so if you do not know where exactly the main region for activations, you can rely on about 500 base pair. And it's much simpler because transcriptional activation usually is the main control mechanism of gene activation. And so when you have a transcription binding, recruiting, or activating transcription, and that's the main step, I would say, for a lot of the regulation. And so as soon as you have a transcription factor binding and activate initiation, transcription initiation, and then RNA polymerase II will just come to the gene and transcribe. So this is a lot of studies have been done in Saccharomyces cerevisiae and Schizosaccharomyces palmi about this. And so because of that, then we can actually measure, if we want to understand a gene regulation, we can identify the promoter region by identifying where transcription factors or transcription cofactors, which are factors that recruit to the DNA by transcription factors to remodel or to modify chromatin. And some of them are actually functioning to recruit RNA polymerase to the promoter. And so if we can find where they bind, then we would be able to know more about their functions and when they are turned on and when they are turned off. And then we can also measure where RNA polymerase II occupies on the genome to understand when they are transcribing a gene or when they are not transcribing a gene. And so we use ChIPseq, this technique a lot to understand the transcription regulation mechanism. And this will become the reason why we use this and why I explain this will become clear in a moment. Okay, so overall, my lab actually uses a functional genomics approach and apply these basic techniques that I have learned in Kevin Stru's lab and apply it to understand basic fungal biology and understand primary, secondary metabolism and understand processes related to infection. And for today's talk, I'm going to present a topic that is related to basic fungal biology and infection. And I don't think I have to convince you how important fungal pathogens or fungal infections are, and many of you are working on fungal pathogens. And I listed here 5 important fungal infections or 5 common fungal infections, and these are caused by fungal pathogens listed here. Cryptococcus neoformans, worked by a number of you, Candida albicans, Aspergillus fumigatus, Histoplasma, and Theromyces malafii. One thing I want to point out here is that among all this, well, for most of them except for Candida albicans, the main infection route is through breathing in spores produced by the fungus. And in other words, the fungal spores are the infectious propagules of many fungal pathogens. And this is really why we were also interested in understanding more about Conidia and understanding more about spores. In addition to being important for fungal infection, the spores are also the source for plant infections as well as food contamination. And as you can see in here, a lot of plants in our food can be contaminated when a spore lands on the food, and then infections can occur. And when foods are contaminated with fungi, and then we have the problem of mycotoxin contamination, for example, aflatoxin, which is toxic and carcinogenic and can cause liver cancer. And so spores are really therefore important to understand, and this is what we were trying to do. One big problem with fungal spores and fungi, as you're all aware, is they are everywhere. They can actually grow on almost anything, including on the wall, and you can see all these black things are actually, the pigments are coming from Conidia or fungal spores. And then fungi can grow on anything, and they are found everywhere, and they produce a large number of spores. And as a result, then in the environment, there are a lot of fungal spores, and it is estimated that we actually breathe in a few hundred spores each day from the environment. And so this is really therefore important for us to understand the spore biology. And so, because my lab work mostly on Aspergillus nidulan, and because of my background, so we actually started this project on Aspergillus nidulans. And another important reason for why we picked this fungus is because the developmental stages of Aspergillus nidulan is very well defined, and we know exactly at what time different stages occurs. We also know the different cell types that a conidiophore, which is the developmental structure for producing spores, if each cell type and roughly what they do. So a lot of research has been given into Aspergillus spore development. And so we thought this is a good model to start. And of course, we also extend our research findings to other filamentous fungi, as I will show you in a moment. But we started off with Aspergillus nidulans. And so what do we know about Aspergillus conidia or Aspergillus development? And this is EM pictures of a conidiophore, and you can see these are conidia, and they are produced in chains, and each conidiophore can have a lot of conidias produced, up to hundreds to thousands of conidia. And sometimes these chains, depending on which species, can be like 20 conidia in a chain, or up to a few hundred conidia. And these conidia are actually designed for dispersal. And I've shown in this sort of cartoon here, taken from the web, that they can be detached when there is wind movement. They can get blown away, and this is a way for how they go and find a new environment for growth. And so Aspergillus conidia are really designed for dispersal because they are light and they are small. And so once they actually go to a new environment, and when the environmental conditions is favorable for them to grow, to germinate, they will break dormancy, and they will just germinate and grow filamentously as hyphae. And when they receive developmental cues for asexual development, then they will put up the conidiophore, the structures of conidiophore, they initiate that, and then produce again chains of conidia to make more of itself. And now this can be spread again to a new environment, and the cycles go on and on and on. What is interesting and important? Why is this? Why are they designed for survival? Is because these conidia are actually known to be very to be stress resistant. They can actually stay dormant and alive for a very long time. And if we sort of keep them in a favorable conditions, proper storage conditions, they can actually stay for 10, 20 years without a problem, and we can revive them after a long period of time. So they have the ability to stay dormant. And so what do we know when we started this project? So there are a number of things that I want to sort of give the background so that you can appreciate the findings from our research. And so first is, as I mentioned, they are multicellular, and they are produced by a conidiophore that are made up of multiple different cell types. And hundreds of conidia can form in each conidiophore and in a chain, and one by one, this is a new conidia form, and then as another conidia is formed, then this get pushed up, and so you have a chain of conidia with the mature ones at the top and the immature ones at the bottom. And it is generally assumed that nutrients and growth conditions are sensed by food cells. These are mother cells, if you would like to refer them as. And the signals are then sort of somehow conveyed or passed on to the Conidia. And so this is generally assumed. And by the way, I sort of marked them, color-coded them for conclusions that have experimental evidence and ones that are generally assumed. And so another important information is that there are a large number of studies that show that Conidia actually contains a large amount of highly stable mRNAs. And these mRNAs can be stable for a year without any change. And these mRNA are thought to be made and produced by the mother cells, which is phyolite here. And they thought that Conidias are really passive and they receive all the materials they need from the mother cells, and including mRNAs, transcripts. They are all pre-made and packaged in Conidia. And therefore, you can see, you can imagine Conidia as a luggage. When you want to go for a travel, you pack everything you need. And same for Conidia, everything is needed for germination, for the sort of next life cycle will be made already, ready in encapsulated in Conidia. So this is again what the field generally assumed. And then dormancy is established when Conidia are formed. So in other words, so these are really passive particles and they are kept in a dormant state. And when they arrive to a new place with favorable conditions, then they will germinate and grow. And so we really started this project as a sort of a curious, sort of a fun project. In fact, this is a side project of my PhD student when she first started, and I asked her to play around with this idea, which is to ask, are the so-called dormant Conidia really dormant? And how dormant are they? And because of our background, because I think of transcription all the time, and the question then I asked was, do Conidia actually have transcription activity? And so and my reasoning is if Conidia are dormant, then they should have minimal, if any, transcription activity. So it was just like one-off experiment, and to us doing a ChIP experiment is actually very simple, and so I asked my student to try that. And so, as I mentioned, we use ChIP a lot. So in this particular experiment, we use an antibodies to pull down RNA polymerase II. And RNA polymerase II, when it's engaged in transcription, it will be associated with DNA and with the chromatin. And when we pull them down, and we can isolate the DNA and then send it for sequencing, whatever DNA that we pull down and get sequenced, that means that that particular region of DNA must be being transcribed by RNA polymerase II. And so when we get the result, we will see from a genome browser view, we will see the signals covering a region, and this region is actually actively transcribed by RNA polymerase II. So this is really the simple experiment that we sort of did at the very beginning when this project begins. And so to our surprise, this is a genome browser view for one chromosome. To our surprise, we actually see a lot of transcription in Conidia. And the amount of transcription is actually comparable to actively growing hyphae. And as you can see from these two tracks here, and this is the input control to show that all these signals are actually real signals. And when we zoom in, we actually do see these signals are covering gene bodies. Again, as I said, this means that they are actively transcribed. And so this was actually very surprising, and not only and we did this not only for Aspergillus nidulan, we also did two other filamentous fungi for Aspergillus fumigatus and Mannaphii as well. And you can see that in all three cases, Conidia has a lot of transcription going on compared to hyphae, which is actively growing, and the levels is about the same. And so that was surprising, and that sort of we did not expect that, but of course, I mean, at the back, I was hoping that this is the case because this become then interesting. And so, but a lot of, because the result is so unexpected, then we want to really make sure that this is not due to contamination or technical artifact, or some people actually, when we present the work, they think that perhaps the polymerase II is just somehow stuck on the DNA, and so this is not really active transcription. So we really have to convince ourselves first, and then also others, that this is not an artifact. So we look at a number of things. So to cut this long story short, so we look at under the microscope to make sure that the starting material that we studied is indeed Conidia or spores. And you can see this is really clean. There is no hyphae material in here. We look at the genes that are expressed, and you can see that all of them are actually very specifically enriched with Conidia function, but not so much for hyphae, and there is nothing for DNA input. And so this suggests to us the genes are really physiologically relevant for conidial development. We also look at transcription activation. Recall my introduction slide that we can actually identify where on which genes are transcriptionally activated by measuring, for example, TF2B, which is TF2B and TBP, which are transcription initiator proteins. And so they are required to put RNA polymerase to the gene. And so we measure transcription activation in both Conidia and hyphae and measure and compare the two cell types. And again, we see very similar levels of transcriptional activation. We also look at hallmarks of active transcription, like depletion of histones at promoters, as well as activation marks. These are histone modifications that are associated with active transcription. So we measure all this, and all this points out to us that there are actually active transcription happening in Conidia. And most importantly, we to make sure that this is not stuck RNA polymerase, we actually measure RNA levels by RNA-seq and compare it with RNAP II ChIPseq, and we can see good correlation, which means that whatever we see RNA occupancy on the DNA, we actually can measure RNA as well. And so this sort of convinces us, more importantly, than convincing other people, because we have to invest a lot more time to study this phenomenon. And so to convince us that Conidia actually have very robust RNA polymerase II transcription activities. And so we also look at RNA polymerase 1 and 3, which are for ribosomal RNA expression and tRNA expression. Again, we do see high levels of this going on in Conidia. Again, it's unexpected. So the first take-home message that I want to give out is that Conidia, unlike what was thought before, which was thought to be passive, they actually have robust transcription activities, and they can synthesize mRNAs, tRNAs, and rRNAs by themselves, rather than by the mother cells and package into the spores. They actually make all this by themselves. So now knowing that there is transcription activities and we are convinced that they are real, so then our next question is, are these transcription events specific for spore development, or they are actually responsive to the environment? And so we did a simple experiment, repeating the whole experiment again, but now this time we include more conditions. We look at conditions that have high salt and low zinc, or we actually change the conidiation temperature to high temperature or low temperature. And then we perform the same PO2 profile, and if this is really for spore development, then we expect when we harvest them at about the same time, we should see about the same transcription programs. Or if it is responsive to the environment, then we will see different programs. And so what we observe is that the Conidia actually can produce highly condition-specific transcription response. As you can see from here, cold shock express this set of genes, and then heat shock Conidia actually express another set of genes. And when we put them on different media, we do see very different transcription profiles. And these are actually real signals covering genes. And these genes, when we look at the functions, they actually are physiologically relevant. And one example is heat shock genes. And when we have heat shock Conidia, in another word, Conidia that have been heat shocked, we actually see all these heat shock protein genes being turned up as compared to cold shock. And for zinc limitation, we can actually grow them in zinc depleted conditions, and we can see all these zinc response genes are being turned up compared to control. And so on and so forth. We actually did a number of different conditions, and we see the same trend, and suggesting to us that Conidia can really make environmental specific response. And so this comes to my second take-home message, which is, again, they are not sort of dominant, but they are responsive to the environment. And and we can observe this at the transcription level. So our next question then becomes, well, if they are active, but we know that they are actually dormant, so when during the developmental program or maturation process, when do Conidia actually enter into dormancy? And so again, we use the same method, but now designing it slightly differently. We actually measure transcription activities at 3 days, 6 days, and 17 days, and ask when do we actually start to see no transcription, which means that when do we actually see Conidia entering into dormancy. And so we did that, and to our surprise again, we actually see transcription going on even 2 weeks after a Conidia has been formed, which is surprising because we thought that, well, when Conidia is formed, they enter into dormancy, but we actually see transcription activities for 17 days later. And there are about 120 genes that are actually being actively transcribed at 17 days. Well, because this is again quite controversial, and we have to again also convince ourselves before we can convince others, we wanted to use a different method to get additional evidence. So we turned to measuring ATP. We reasoned that if the cells are not dormant and if they are still sort of active, as we have shown for transcription, and transcription is actually very highly energy demanding, and we expect that Conidia, having not much resources, they are not growing actively, they would not produce ATPs, and therefore, if transcription is consuming ATPs, then we would actually see depletion of ATP over time. And so this is what we measure, and we go and measure over a long period of time and ask how much ATP is remaining. If Conidia are dormant, we expect them to sort of have a same level, roughly the same level over time. If they are very active, we expect them to deplete, just like what I've shown. And so this research suggests to us that, yes, not only that we do see transcription activities, but perhaps other cellular activities are pretty much active, so that ATP levels sort of deplete over time, until the point that 50 to 60 days, we actually see not much transcription left. And so you may wonder what happened to the viability. And in fact, when they actually reach 50 to 60, 70 days, when there is no ATP, those spores actually start to die as well. And in other words, so as soon as they deplete their ATPs, they are not viable. And we think the reason for the inviability is because the germination process actually requires energy as well. And so if there is not much energy left in the Conidia, then they would not be able to initiate growth, initiate germination, and therefore they lose viability. So this comes to sort of, again, we thought this is strange. I'm telling you that Conidia are not dormant yet, and they actually will die eventually because they do not stay dormant and they consume their ATPs. But then we actually know this is a fact, that Aspergillus Conidia can actually stay dormant. I told you that we can keep them for 20 years and we can still revive them, they can stay alive, and then what's going on? And so we went back to think more about our experimental procedure. We actually perform our chip on Conidia that are attached to conidiophores, just like this. But when we actually store our Conidia, we usually separate them. And so we wonder whether or not the point that Conidia enter into dormancy is related to whether or not the Conidia are separated from conidiophore, which is the mother structure. And so if that's the case, then maybe we can do an experiment to test that. And so that's exactly what we tried. So we performed the same experiment. We keep Conidia on a plate, which is on a conidiophore, on the mother structure for 3 days, 6 days, and 17 days. And we added one experiment, which is we separate the spore, we harvest the spore, and then we keep them at the same condition for another 2 weeks, up to 17 days. And then now we measure ATP levels and ask whether or not this will actually put Conidia into dormancy. And as you can see here, when we separate them at 3 days and keep them for another 14 days, nothing much has changed. ATP level did not deplete, suggesting to us that it is this separation that put Conidia into dormancy, which means that all the cellular activities sort of stopped. And so we use another methods to verify that. So we went by to look at molecularly and identify a protein that are degraded over time during maturation. And we separated the Conidia, and then we see that the degradation does not happen, so that we actually sort of prevented the turnover of wet A by separating them, suggesting that cellular activities can be stopped once we separate Conidia. We also measure mRNA levels by RNA sequencing, and this is a 3-day-old Conidia, and we separate them and then keep them for another 2 weeks and measure mRNA again, and you can see the correlation is very high, suggesting that they did not change up over the two weeks period. Again, suggesting that separation actually put Conidia into dormancy. Now, well, when we do the separation, there are two things that happen. One is the separation. Another thing is actually dehydration, because we cannot keep Conidia sort of not dehydrated. We have to keep them on a filter paper and then keep it for 2 weeks. So we ask whether it's the separation that put Conidia into dormancy, or it is a separation. And so we did an experiment by separating the Conidia with and without putting them in water, and then we treat them to heat shock. And so if they are responsive, if they are not dormant, they would be able to respond. And as you can see, this orange bar, when they are separated but still not dehydrated, they can still actually respond, suggesting that it is actually the dehydration that stops cellular activities in a way that induces dormancy of Conidia. And so, we think that yes, dormancy still happens in Conidia, and it actually happens upon dehydration. Okay, so this is as what I said. The third take-home message is that dormancy actually established during dehydration. I noticed that I'm running out of time, so I will try to speed up. But this is actually important. It raises an important question, because I show you that Conidia actually consume ATP and they can die from it. Okay, they can lose viability without ATPs, and then it makes us wonder, why do they want to keep transcription functions and risk their life for staying active? And that suggests to us that perhaps the transcription must be very important for the survival of the fungus. And so this leads us to sort of address what is Conidial transcript function, and there are two main ideas in the field, not very well defined. One is for development and maturation, and one is for germination. And so we went back to our data and tried to find experimental evidence to suggest whether the transcripts are important for survival and which aspect of the survival. And so to cut the long story short, we actually have identified evidence to suggest that it is related to development and maturation. As you can see that when Conidia are developed under different conditions, they are actually producing different proteins, suggesting that transcripts are made into proteins for development and for maturation. And we can see even Conidia expressing different levels of sugar transporters. Similarly, we can see secondary metabolites. This is actually an interesting example because glyotoxin is known to be an immunosuppressant, and Aspergillus fumigatus is a pathogen. And so having a high level, we actually detected this cluster genes expression at a higher level in zinc-derived, zinc-starved Conidia. And we measure the glyotoxin level in Conidia, in these two types Conidia, and we actually did actually detect the glyotoxin, suggesting that zinc-starved Conidia may actually have more of this immunosuppressant chemicals compared to a control Conidia. This is interesting from the perspective of infection, because Conidia with or without this immunosuppressant may actually have very different outcome in infection. So we actually detected, we tried to sort of study many different factors and try to identify, correlate mRNA levels with protein levels. To our surprise, we actually see a good number of proteins that cannot be detected, even though we do see their mRNAs, suggesting to us that this may be important for germination. So they are not used in Conidia development, but they are used for germination. So this led us to sort of pick one example to test whether or not this is really the case, and we actually now looking at germination time points, 2 hours, 3 hours to 7 hours after germination, and we have picked one example, which is SAP-A, which is important for zinc limitation response, and we observed this particular gene having a higher expression in zinc staph Conidia compared to control, and this higher level is translated to an accelerated expression of SAP-A protein during germination. In another word, the spores are already making an mRNA that is not used in the spore because we do not see any protein, but it is used to speed up the response in the absence of zinc later during germination. And as a result of this accelerated production of SAP-A transcription factors, we actually also see accelerated response of downstream genes, which are zinc transporters genes as shown in here. In all three cases, we see zinc staph Conidia actually turn on these transporters much quicker than control Conidia, suggesting that this transcript of this transcription factor is gearing towards the germination stage. It's used for germination rather than for development of Conidia. And so we then go and look at transcription factors, genes in different Conidia, and we see a highly heterogeneous expression pattern among these 61 transcription factors. And I have shown one example for SAP-A, and if all these 61s are actually behaving in the same manner as SAP-A, you can imagine how heterogeneous the response would be, because these transcription factors will turn on a whole set of different genes depending on whether or not they are activated in a specific Conidia. And so something that I did not mention in the introduction is that there are a lot of studies that shows even though Conidia are produced from a genetically identical mother cell, and they are all genetically identical, they actually have phenotypic heterogeneity. All these can have different surface proteins, they have germination behavior, and they can have different variants ability, depending on where they are developed from. And so this is not only shown in Aspergillus, but it has actually been shown in many other fungi. And the earliest paper that I can find is about 60, 70 years old. And so this is a long-standing question, and how a genetically identical Conidia can produce phenotypically heterogeneous Conidia population. And so we thought perhaps our Conidia transcription, the transcription that we observed before dormancy, may be the reason for this heterogeneity. And so if this is the case, we would be able to identify from our experimental data to find examples to demonstrate this point. So we went back and looked at all the gene transcription expression profiles, and then identified genes that are relevant for specific physiology, like hydrogen peroxide resistance. We can see in sodium chloride treated Conidia, we can see genes that are relevant for hydrogen peroxide turn up, and we can see increased resistance. Same thing for fluconazole. We can see genes that are related to fluconazole resistance turn up in this Conidia, and then we can see resistance. Same for glyotoxin production for Aspergillus fumigatus, and so on and so forth. We have a number of examples to demonstrate that, suggesting that, again, the transcription that we observe in Conidia are actually physiologically important, and they can contribute to the heterogeneity after germination. And because they have very different properties, we also measure whether or not different Conidia or Conidia, genetically identical Conidia developed from different conditions, can also have different variance abilities. As you can see from this Galeria infection experiment, we actually do see variations. They all still killed, but the kinetics of killing is actually very different. And I have shown you this just now, that the genes for glyotoxin cluster are turned up in zinc staph. And so we thought, well, what is the significance of this, and is this also affecting secondary metabolism production later in the growth? And for your information, a lot of these secondary metabolism are actually producing many days later after germination. And we actually sort of wonder whether or not transcripts in Conidia can affect a process that is a few days later. And so this is the experiment to show that indeed those transcripts can affect the process even 5 days later. And you can see that the levels turn up many times. We actually have an example that can increase by 10 times. And so whatever that is made already in Conidia can affect the capacity or the ability of the fungus even many days later after germination. And so, well, that all these observations are sort of associations, and we really want to make sure that, or we want to demonstrate that Conidia transcript is the mechanism for heterogeneity. And so we artificially express, and we artificially express a transcription factor in Conidia and ask whether or not we can completely change the physiology of the Conidia after germination. And so we pick one secondary metabolite transcription factors and overexpress it in Conidia artificially, and then take the two Conidia, which is non-induced and induced, and put them under the same growth conditions, and then measure the secondary metabolite levels. And as you can see, there is a 10 times difference in Conidia that are already carrying the transcription factors. In another word, this is like the SAP-A situation. It is actually priming the Conidia to produce, to increase the capacity of the secondary metabolite production. And so with this experiment, we thought we are quite convinced, and we also convinced the reviewers that this is really a case that Conidia transcripts is the cause for phenotypic heterogeneity among Conidia. And so the last take-home message is that I have shown Conidia transcripts actually play very important roles, unlike previously recognized, and they can affect development, they can affect maturations, they can affect germination, as well as metabolism that happens 4 or 5 days later. And as I mentioned, we propose that the heterogeneity is actually caused by Conidia transcription before dormancy. And this is a schematic model that we put in the paper, and if you're interested for more detail, I actually refer you to this paper. But briefly, what we wanted to say is that Conidia actually transcribe the relevant genes according to the environment. And so these two Conidia are developed under different conditions, and so they may make different proteins like transporters and cell surface proteins, and this making more the red and this making more the blue. And these transporters actually are physiologically different, and they when they germinate, they will import, like for the case of transporters, they will import different nutrients, and this one imports the red one better than this one, and this one imports another set of nutrients, and as a result they actually would behave very differently under the same conditions. And we think that this is a way for how Conidia actually prime themselves, prepare themselves for the environment. And all these Conidia transcripts can also have effects later, subsequent growth, as we've shown for secondary metabolites. So this is the general model that we put forward to explain. And so ultimately, so this is why we think fungal spores actually proactively prepare for their future before entering dormancy. And we had a lot of fun with this project, and so I got a niece to actually draw, who is very good at drawing cartoon, and I asked her to draw a cartoon for me. And this is what we think when a Conidia is developed under hot environment, the Conidia equip itself with everything is needed to survive under the environment. And then similarly, then the Conidia will equip itself to grow under another condition, like in this case, cold. They actually both can survive under both conditions. Is whether or not they survive better or not, rather than this cannot survive here. It's actually they can survive. It's just a matter of time, the kinetics, how well they grow, how fast they grow under the conditions. So in a way, this is sort of a mechanism to give a competitive edge to Conidia, rather than to give it a yes or no type of growing. Why is this important? I told you before that, well, fungal spores, as we all know, are everywhere. We're breathing fungal spores every day, hundreds of them. But all these spores actually comes from very different environment. Spores in Macau can be very different from spores in Australia. Even spores in Sydney can be very different from spores in Melbourne, because the environment that they develop from are very different. And so we think that this is relevant to the infection sort of situation, because we already know that there are a lot of genetic differences. There are a lot of selections to select certain genotypes, certain genes in certain isolates, but we are adding into it that spores, when they develop, they add additional levels of heterogeneity with respect to the mRNA contents, the ATP levels, the maturation states, etc., etc. And all these can make the whole thing highly complicated. And we think that we have to really pay attention to not just the genetic differences, but how we actually generate spores and what are actually the conditions that make Conidia more pathogenic than others. So this is sort of what our findings sort of make us think a lot more in this regard. So with this, I really want to thank the people who did the work. I'm sorry for running over time because I initially planned for 50 minutes. Anyway, so Wang Fang is a PhD student, graduated. Most of the work done by her, a very hardworking, talented student. And with help with from Puja, all the bioinformatics sort of analysis were initially taught by Puja to Fang Fang, and Fang Fang later on pick up all the skills. And now, in a way, you can think Puja is the teacher to Fang Fang. Fang Fang now is very independent in both wet and dry analysis. And so she was helped by people who are highlighted in blue in here. And this is my previous lab. My lab has sort of downsized a little bit because I had too many people and I really need to help them graduate, so I stopped hiring for a while. So this is my people now. Funding, this work cannot be finished without funding support from the faculty, from our university, as well as our funding agent in Macau. The main funding source is our casino. So the revenues, some of the revenues goes to our funding, and so I welcome all of you to visit Macau. If you go to Hong Kong and go to China, please come to contribute to my funding source, or you can double your funding amount by going to some of the casinos here. So with this, I would like to thank you for your attention, and I'm again sorry for running a little bit over time, and I'm more than happy to take questions.
Wow, that was an amazing talk, Chris. Thank you so much. Very, very comprehensive, very convincing. I can see now why it was published in Nature Microbiology.
Thank you.
Yeah, no, great. Are there any questions from the audience? Dee's got a question?
Yes.
We're on mute, Dee.
Thanks,
there you go.
Thanks, Chris. That was a really fascinating, wonderful talk.
Thank you.
I don't know if you know Arturo Casadevall, but he talks a lot about the possibility of climate change and increased temperatures potentially making fungi more virulent. And I guess listening to your talk makes me worried that that's really the case. As the temperatures get higher, closer to our body temperatures, that the spores could be getting more and more well adapted to affect us. I was just wondering what your thoughts are on that.
Well, based on we really want to sort of move this into mouse experiments, but we are not set up in animal studies, and I think that should be the next step to test how important is this phenomenon. But based on our Galarian model, we think that it actually helps it to infect a bit faster. The kinetics is both different types of Conidia will kill eventually, but it's whether or not making it faster or slower. It's really difficult to know how it plays in a host because that maybe 5-10 minutes or maybe half a day difference could mean a lot in terms of macrophage response, host immune response, but this is really not clear. And it will be really nice to put it into a mouse model and see how much it contributes. So without that result, it's really difficult. Of course, I hope that it would be an important factor from the research perspective, because this is a great finding. But I hope this is not the case, because as you say, global warming will make them more adapted and then be more a problem to human health.
Absolutely.
Yes.
Yeah. Thank you.
Yeah, I agree. I think it would be great to do the studies in mice, because I mean the glaria are a good tool for getting an idea about whether something's more or less virulent. But I mean, I find generally with Cryptococcus, when I'm making mutants, the correlation is not as good as it could be. So yeah, it would be really interesting to sort of have a look at some of these studies in a mouse model. And the other thing I was going to ask too is these are transporters that get upregulated. Do you know whether they also are involved in effluxing drugs, and that's potentially that could contribute to antifungal drug resistance?
Yeah, that's a good question. So we try to look at it very hard. We have a lot of we have all the transcription data, so we actually look at this very closely to separate all different functions and find ones that are relevant for drugs. And we do see sodium chloride actually turn on some drug transporters. And so we tested on fluconazole, and indeed we actually see a bit of sort of more resistant to fluconazole. So I'm sure it plays out in the spore levels. And we do have experiments to show that if we artificially overexpress some specific transporters, we can make this Conidia grow faster than when it does not have those transporter turn on. And we have also looked at, and this is actually also important in terms of thinking about experimental setup, we have looked at mutants. We always make a deletion mutants, and then we throw and we compare a phenotype, and we see some of the phenotypes are actually due to changes in the Conidia transcription profiles, rather than a real direct effect after germination. In other words, we do see mutants that changes the spore contents, changes the transporters on Conidia, and as a result, the Conidia can grow faster or slower, and then it make the Conidia grow faster or slower, and affecting a lot other subsequent physiological pathways. And so we do see a lot of this, and so in another word, we can translate whatever we see in the transcription to physiology.
Right. Okay. I noticed you had a lot of glucose transporters being upregulated too, and I think there's precedent in the literature that they can also pump out antifungals as well.
Oh, okay.
Yeah, I think we did a study with an anti-parasitic drug called miltefosine, and there were definitely glucose transporter proteins that were involved in conveying resistance to that particular drug.
I see. Yeah. Well, other than glucose transporters, I think the cell surface proteins are actually also interesting, because we do see them change between different Conidia preparations, and those are the surface proteins that are recognized by our immune cells. And so that can actually play a role, and we do see aflatoxin, which is again pointing to us that different Conidia can greatly affect how the Conidia behave in the host, and that was actually surprising to us as well.
Yeah, right. Any other questions from the audience? If not, I just had one more. Look, it's a basic science one, and it's really all about metabolism. So I know that you looked at ATP levels throughout the maturation process. And I mean, as you know, ATP can be generated rapidly through fermentation, or, the Conidia could potentially engage the mitochondria and make a lot more. And I just wondered if you'd done any metabolic studies to determine, the level of metabolic activity in terms of fermentation and oxidative phosphorylation.
No, we haven't done any. And in fact, there are papers to suggest that in Conidia, there are really minimal metabolic activities. And that's really the basis for why we sort of had the idea of measuring ATPs, because we think that it's not generating ATPs. And if it's
Right.
active, it will consume. And so over time, it will just have a net depletion. And so
Okay.
no, we haven't.
Okay, so you still need energy to make all the RNAs though. And I guess that could probably be made through the pentose phosphate pathway, which is really sort of like a parallel pathway to fermentation. So perhaps the mitochondria are not even needed. It would be fair enough to say to generate that arsenal of RNAs.
Yep.
Yeah. Amazing. That's
Thank you.
great.
Thank you.
Any final questions before we wrap up today?
I don't think so.
No.
But thank you for that
Okay.
talk. It was super interesting.
Thank you. Thank you for having me.
Right. Okay, well, thank you again for your time and your presentation. And yeah, and I hope you don't go into any more lockdowns, Chris.
I hope so too.
Yeah, so we wish you all the best and look forward to seeing you again, hopefully at a conference in the future.
Yeah, I look forward to visit Australia.
Well, that's
Right.
good. So
We'll
we'll
see you
have to
then.
invite you to come
Yeah.
out.
See you.
Yeah.
Okay,
Thank you.
thank you very much. See you
Thank
later,
you.
everybody. Bye-bye.
Bye-bye.
Thanks, Chris. Thanks, everyone.
Thank you.
See ya.